Okay, yeah. Good morning, everybody. So the title of my talk is Why Bother About Microbes in the Underground? I must say, geoscientists usually divide their colleagues up into two categories. It's those that deal with sediments, and it's those that deal with igneous rocks. And so as the igneous rocks are harder, they're called hard rocks. And so the people deal with this, they're hard rockers, and the other ones are soft rockers. So you just heard a talk by a die-hard hard rocker, and I must say, I'm a soft rocker. I don't deal with igneous rocks. I'll just stay in the sediment. So no more about basalt or any kind of other. It's pretty cool stuff, but I just stay with my soft rocks. So why do we actually bother with microbes in the ground? So there's a number of reasons. So there's a number of scientific reasons, and there's also societal and economical reasons why we deal with this. And one is from a scientific perspective. The ecosystem in the subsurface biosphere is the largest continuous ecosystem in the world. When we think about the sediments on the sea floor, these are grains sitting there, but there's pore space. So there's always a pore between these, and all these pores are interconnected. So there are very few spots in the world's ocean where we don't have any sediment because there is some mid-ocean ridges or so. But other than that, it's a huge system where all these pores are interconnected. So this is an ecosystem that stretches once around the globe, and depending where you are, it might be tens of kilometers thick. So this is a gigantic ecosystem, not just in areal extent, but also in thickness. And I would like you to think about it as a huge continuous system that changes with depth, also geographically, but it's all interconnected. And over long geologic periods, there is interconnection between these different environments. So it's also one of the least explored ecosystems on Earth, as we already heard from Jason. So we only have a couple of pinpricks on the sea floor where we have any kind of microbiological analysis from. So here is actually a map where you can see that all these points are where we have actual activity measurements. So where people have used radio tracers or other techniques to measure microbial activity to show, yes, there is really microbes, active microbes down there. And so as you know, there are only two drill ships out there. There's a second platform, a third platform that might be used every once in a while. So chances of getting out there are really, really scarce. And I must say with some pride, of all these numbers, all these ones here are actually mine. So there's not much chance of actually getting out there, getting pristine sample material in order to get those experiments done. And so this plot took about, I would say, 20 years of work to get these few numbers. Another, I would say perhaps the biggest driver of us is that the presence of active microbes deep in these environments really challenges our concepts of the ultimate limits of life. Seriously, even after decades of research, in many cases, we just cannot explain why they are there. Because they should have died. They should have just starved years ago, or actually millennia or millions of years ago. And this was from the South Pacific Gyre, so this area really, really far. It's the most remote place on the world's ocean. You can't go any further away from dry land than there. And it's also the most oligotrophic, the most nutrient-depleted place on Earth. And still, when we drill into the sea floor, we find cells. Not a lot, about 1,000 to 10,000 cells per cubic centimeter. In a coastal surface sediment, you find millions to billions of cells. So it's definitely orders of magnitude lower, but they are there. They are there and they're alive. And first we thought, where do they live from? Because when we went out there, we couldn't even measure any organic matter because the organic matter content in the sediment is so low. It's 0.0 and a little bit percent. So the normal techniques that we have don't even measure that. So we had to bring special machines out there to measure it properly. But still, they're living off of it. From an economical standpoint, one of the biggest drivers is actually hydrocarbons. So there's all this talk about peak oil, whether we've reached peak oil or not. I would say we haven't reached peak oil, but we've reached peak cheap oil. Everybody wants the super light, sweet crude oil like this. This is like an Arabian oil that basically it comes out of the well, you can put it in your tank. That's like the easiest material you can use. It's easy to refine, huge yields, very easy to work with, but that's about to run out. All the new wells that are being explored right now, they deliver usually much, much less quality in terms of oil, or actually it's biodegraded oils, and they look like this. This is pitch black stuff, and this oil has been biodegraded, so microbes had to go on this, and all the nice stuff, the valuable stuff, they have munched up. So we're left with that, and refining that material takes much more energy, much more time. And so we still haven't gotten a good understanding how microbes degrade oil, and why are they doing it in certain cases, why not in others, and what kind of processes are there. And this is really a big, big driver to understand how we can make better use of our natural resources that we have, and how can we economically exploit them much better. Not so much in Germany anymore, because there was the topic was basically banned here. But in other countries, it's still a major topic, is CO2 sequestration, so storage of carbon dioxide underground. And when you think about it, sometimes they pump it into deep aquifers, into other kind of reservoirs, depleted oil reservoirs. When you start pumping thousands to millions of tons of liquid CO2, which is an ultra-reactive species, if you pump it down there, you really start altering an ecosystem to such a major extent. And still, so there, we know there are microbes down there, they do something, and suddenly you add a solvent that does some very interesting chemistry down there, especially when it's not a pure solvent, but mixed with a bit of hydrogen sulfide, a bit of sulfur dioxide, and so on. You decrease your pH down to, let's say, 1.5. You really start altering an entire ecosystem. And we think, well, we pump this stuff down there, it stays down there for geologic timescales, but we don't know what the microbes are doing down there. So we should perhaps get a handle on that prior to doing this. It was already mentioned, arsenic is, for instance, also a major point. So groundwater is something, it's full with microbes. It's not a bad thing necessarily, but in some areas it is. For instance, in Southeast Asia, we have major, major issues with arsenic in groundwater. I've just heard in this talk earlier today that this is also an issue in the US in some areas, but from what I heard, it's definitely not as bad as in Southeast Asia, where you might have wells that are completely loaded with arsenic, and arsenicosis is a very, very prominent disease there, and about 500 million people are at risk of being poisoned by arsenic in Southeast Asia. And arsenicosis is a really, really nasty disease, because basically your toes and fingers are just rotting away on your body. So, and this is a huge societal need. And so you might have two wells just a few hundred meters apart. One is loaded with arsenic, the other one is pristine. That's all microbially controlled. But so far, we haven't really understood what are the control factors and how we can perhaps remedy that. So this is all deep biosphere research, but how did it start? How did people think about microbes in the subsurface? And it started pretty early. In 1955, the famous microbiologist Claude Sobel, he went out to the Central Pacific and took a long core, and he developed the first sterile sampling techniques, because when this core comes up and you want to do some microbiology with it, you have to avoid introducing foreign microbes from the surface. You really only want to work with your microbes down from the core. And so he developed those techniques, and he just took small sterile samples, smeared them on an agar plate, and incubated them. And the first sample, he could not cultivate anything on it, was from 7.47 meters depth. And so he concluded that at 7.47 meters, it's the end of the biosphere. Nothing lives below this because he can't cultivate it. So he wasn't just wrong, he was very precisely wrong about this. But he already asked a very, very important question, because he said these sediments, because of their extremely low sedimentation rate, these sediments are hundreds of thousands to millions of years old. How on earth can they survive? Because they have been away from the surface for such long periods. How do they survive? And that was really something that this question he couldn't answer, but this question is bothering us today, still over half a century later. So, and then 30 years, not much happened about it. And then in 1985, Joe Whelan went out on a drill ship. It was the last cruise of the first scientific drill ship, the Glomar Challenger, and they did radio tracer experiments. So they added radioisotopes into sediment cores and could measure production of hydrogen sulfide, of methane from radioactive substrates that they added. So they could show this is only possible through microbial activity. Before, these measurements were not allowed on the ship because when you work with, especially with 14C, 14C is also used for dating cores. But they measure the natural abundance of 14C, whereas they introduce pure 14C. So if there's just the tiniest bit of contamination, they would shoot all the ages into the future of the sediment. And they were so concerned that they would contaminate the whole ship. So this was the very last cruise of that ship. After that cruise, it went to the junkyard. So they said, we don't bother anymore, do whatever you like. And so this was the first time they could use it. Now we use it routinely, but we have a special radioisotope, usually in the far back of the ship, somewhere in a container. They really don't like us. So after this, more people got involved into this topic and said, okay, how many microbes are down there? And so John Parks collected a number of cores from other cruises and did bacterial counts. Actually, his assistant, Barry, corrected all the counts. And in 1994, he published this famous paper and could show that there are viable marine bacterial populations down to at least 500 meters. And that was basically, and he used the term deep bacterial biosphere, deep biosphere. That was the first time this term was ever used. And this gentleman just coined this term, John Parks. There was another 10 years after that it was finally proven that these cells are actually alive by using cardfish and molecular techniques, and they could show that these cells contain ribosomal RNA, which is a clear indicator that these are living cells. And they could show that at least 30% of all detected cells are metabolically active. We know now that there are much more active cells down there, but this was in 2005 or 2003 when this analysis was done. That was about as good as the techniques were. So, and based on the data of Parks and mostly of Parks, there was a famous paper published in PNAS actually in the late 90s by Barney Whitman, and he just did a wonderful literature review. He looked at all the bacterial numbers that were presented from all environments all over the globe, and he made this huge literature study, and he just summed them up. Pretty basic stuff, but very ingenious. And he could figure out that 92% of all prokaryotes, so bacteria and archaea, live in the subsurface, either in the deep marine or the deep terrestrial subsurface, but only very, very few cells live in the aquatic environment, so in the open water or in soils. And then by using a conversion factor of the amount of carbon per cell, he said this is the living carbon. This is the carbon that actually does something in our environment because it's alive. And he just used this factor and figured out how much living carbon is stored in the deep subsurface. And then, of course, the biggest part of living carbon is today stored in land plants, but 40% is stored in the deep biosphere. And so for about 15 years, this paper was cited as a first citation in every single deep biosphere paper because it says, hey, we have little to no knowledge about 40% of the global biomass. It was so easy. It was the best justification you could have. But of course, the biggest driver of this number was the deep marine biosphere. But over the years, more and more cruises were scheduled and cells were counted. And I've developed a technique to actually reduce the detection limit of this method. And so more and more people went out. And so we got more data coming in. And eventually we said, wait a minute. And so in 2012, we published a paper that Jason already mentioned, that we reassessed the global marine sedimentary deep biosphere, not the hard rocks. And so we decreased that number by about an order of magnitude, because before we only had these black counts that all came from, let's say, coastal environments or very organic rich environments. And all the other ones from the mid-ocean gyres, they were just not accessible before. And I always try to explain it to people. Just think about you're an alien and you send drones on Earth. And each drone takes one picture. And then they fly back and you see the pictures and you try to make you try to get an idea of how the planet looks. And you would think, you get 5 pictures, one from Times Square, one from Piccadilly Circus, one from the rainforest, one from Beijing, and one from Hong Kong or Tokyo. You would get a much, much different picture. These were basically the black dots, lots of life. And then you would send 5 more drones, and you would send them to the Gobi Desert, to the Sahara, to the tundra, to the Arctic, and the Antarctic. You would think that's a different planet. And so this is where we could show that there's much, much larger variability in the system. We could use this to build a better model. And so we reduced the number of microbes on Earth by about 50 to 75%, so about two-thirds. So I got this email from a friend saying, "Oh, you bloody Germans, you killed one-third of all life on planet Earth." I said, "No, it wasn't there to begin with." But yeah, he wouldn't want to hear that. Okay, yeah, it's Germans. Yeah, well, yeah. So in my PhD student, Rishi, at this time, he's from Nepal. When I hired him, I said, "Have you ever been out on the ocean?" He said, "Oh yeah, I took the ferry from Cali to Dover. Anything longer?" And he said, Nepal doesn't have much of a coastline. But he did this amazing work, and he sat down and measured the individual length and width of microbial cells in our samples, hundreds and hundreds of individual cells. And he calculated biovolumes, and then we could use we figured out these cells are much, much smaller. Kelly will talk about this later. These cells are much, much smaller than the actual cells that you find at the surface, because these cells are basically starving down there. So this is where we said, okay, we have much less carbon per cell. And this really brought down the number quite a bit. So the total living biomass was reduced by about 1/3 by this. So how deep can we go? Jason already mentioned in the marine realm, the record right now is through drilling. That's actually a picture from that famous Chick-U cruise where they drilled to 2.5 kilometers, where they still found life. It's just, no matter how deep you drill, as long as temperature is still in the biological range, I come to this, you find life. Still basic, it's the same almost in the terrestrial environment. So in terrestrial environments, we have one advantage, we have deep mines. So we can actually go down there. We don't have to drill 3 kilometers. So the mining companies built these large mines, especially in South Africa, and you can just take the elevator down and then just drill horizontally and try to tap something. And it's really fabulous, so that you can go down there, and it's a pretty, it's my colleague Dwayne Mosier at 3 and a bit kilometers, so it's a bit warm down there. Average air temperature is about 55 degrees to 60 degrees, so it's a pretty tough working environment. In order to pass a test to get down there, they put you on one of these home trainers they have set up in a sauna, and they let you run this thing for an hour. Yep. So, but if you collapse down in the mine, to get you up 3 kilometers is an undertaking. So they really want to make sure that these people are in good training. So basically, wherever you look, you find life. I've already talked about temperature limits. So we know the temperature limit, the absolute temperature limit for life is somewhere in the 120 Celsius range. There's always somebody has half a degree more here or half a degree more there or so. But it's about 120 degrees. But this is only what you find in hydrothermal systems, in these famous black smokers, like where these hydrogen sulfide and metal-loaded, ultra-reducing fluids shoot out into the completely oxygen-saturated seawater. But this is an ultra-high energy environment, because you've got all this chemical energy coming up. You have these very reduced species and oxygen in the seawater. So these microbes can just do oxidation reaction and gain energy from this. So at such high temperature, you invest a lot of energy as a microbe to repair all the damage that is produced through the high temperature. And so this is something where you can withstand such high temperatures, but you need lots and lots of energy. Whereas if you go into the deep biosphere, and the sedimentary systems, also in hard rock systems, it's an energy-starved system. So your biggest challenge is not to starve, and you don't have enough energy to repair yourself properly. And this is where we say that basically what we've seen is that if you reach about 80 degrees, this is where the fun stops. So in sedimentary environments, it's usually 80 to 85 degrees is where you run out of microbial life. This is very interesting for the hydrocarbon industry, because once an oil field was heated to over 80 degrees, it does not degrade, even if it's now emplaced at a shallower depth where the temperatures are lower. Still, you have pristine oils, but you have to pasteurize the field once to over 80 degrees. So this is actually called paleopasteurization. So pressure is not much of a limit here, actually, because they've done experiments. They even put poor little cells and diamond anvil cells and put them at the pressure that you find in the Earth's core. And still they survive that. Not for extended periods, they might shut down their metabolism, but pressure is not an issue. Of course, for our equipment, pressure is a major issue. And you all know these ugly styrofoam cups you get in American hotels for your coffee. This is when you take one of those cups and put it down on the seafloor. That's what comes up. And just give it a round. We tied one to a core and send it down. As a little, yeah. It's also when you have these styrofoam heads from wig, for wigs or puppets or so, and you send down, you get these tiny shrink heads back. I couldn't find mine, unfortunately. Yeah. But the deep biosphere is also a refuge. So we've got this wonderful drilling through the Chesapeake Bay impact crater. 25 million years ago, a huge meteorite hit here. And what we found was that when the meteorite hit 25 million years ago, it excavated a hole that was about 1 kilometer deep. So it just blasted off the material. But then, a couple of minutes later, there was a back surge, and all the material that was thrown into the air and partially melted fell back down. So we got about 700 meters of sediment deposited here that is full with melt fragments. So it was effectively sterilized. And now we measured cell counts, and what we see is that at the surface, down to about 400 meters, we have cell counts that we expect, and then they drop off rapidly, and then we have this long section here where we don't have any cells. So the cells are now recolonizing the sterilized environment. So this is a 25 million year running experiment that still hasn't reached its end. Interesting thing, there's a huge granite block here, 200 meters thick, and below the granite block, we have microbial cells in the same abundance as above. So they haven't even bothered that there was a meteorite hitting them. So even if conditions at the surface get really, really ugly, if you're deep enough, you don't bother. Have we caught the whole story? And actually this is not. So far we've only counted cells, microbes, we counted bacteria, we counted archaea. And a couple of years back, people started thinking about, what about viruses? So in a normal ocean or a coastal sediment, you find about 10 viruses per microbial cell. That's about the average ratio you find. And one virus weighs about 1/70th of a microbe. So when we go deeper into the sediment and we go to very old, very nutrient-poor sediments, what we see here is suddenly the ratio goes up. So there are much, much more viruses than there are cells. And so if you breach this 70 ratio line, that means there's more biomass stored in viruses than there is in cells. And in these very deep, very old South Pacific gyre sediments, we find up to 220 times more viruses than cells. So that means 3 times more biomass in viruses than in cells. The question is, what are these viruses doing down there? Are they controlling the ecosystem? Are they the predators? But why do you need so many of those? Or are they the lions, or are they the antelopes? Are they the food source? Because viruses contain so much beautiful DNA. And DNA is rich in nitrogen and phosphorus. So they're a perfect nutrient source. Then the question is, why aren't they utilized? Why are they just hanging around? So sometimes people think they might be just the predatory antelopes, so antelopes with giant jaws or something like this, that they can do both, but this is something we still don't know, and this is still ongoing research. So there's still lots and lots of questions going on here, and we just don't know where to start, actually. So lots of open questions. Thanks.